going to be talking today about my research related to dissolved oxygen levels in coastal systems in general, but specifically in Chesapeake Bay. And these dissolved oxygen concentrations that we're interested in are primarily the low concentrations, which we refer to as hypoxia. It's a term that we borrowed from the medical community to describe low oxygen concentrations in the bloodstream. There's a number of things that control dissolved oxygen. One of the main factors, ultimately, these microscopic algae that grow in the water column. And at some point, they sort of end their life cycle and end up sinking. And when they sink, they go to the deeper waters of the bay and the bay sediments. And when those dead algal cells are broken down, and it's not necessarily all dead algal cells, can also be material that's washed in with the rivers into the bay. The process by which that decomposes consumes oxygen. And so what controls the amount of phytoplankton, or these microscopic algae in the bay, is related to the amount of nutrients that are available to support their growth. The amount of nutrients that are available to support their growth are related to how much comes in off the watershed. The more nutrients you have, the more of these microscopic plants you tend to have, the more they sink and the more oxygen they consume. If you look around the world, there's these low oxygen areas that are actually fairly common at this point. And it just so happens that where you have high human impact on the land and therefore high nutrient loading into the system, you also tend to have these low oxygen areas. So one of the interesting features about this problem of low oxygen is that there's all these connections in both the production of hypoxic volumes and what their effects are in the ecosystem and how they feed back in to society. And that's one of the things I really think is interesting about this problem. So for example, as I mentioned before, where you have activities on land resulting in high inputs of nutrients to a place like Chesapeake Bay, you tend to have higher volumes of this low oxygen water and therefore less habitat for a lot of the organisms that it turns out we like to harvest and eat. And so there's a negative consequence of the things we do on land, how they affect the water and how that water affects the ability for a lot of different species to maintain high population levels. Well, in Chesapeake Bay, we know there's been documented low oxygen water back until the 19 teens. So in the Potomac River in 1917, there were measurements of what we would now call hypoxic water. In the main stem of Chesapeake Bay in the 1930s, there were direct measurements of what we would call hypoxic water. So well before there were massive amounts of nutrients leaving the land and coming into Chesapeake Bay, there were hypoxic conditions. As one of my colleagues once said perfectly, Chesapeake Bay is an estuary that kind of naturally wants to be hypoxic, and all we have to do is push on it a little bit and it will land itself in a hypoxic state. One of the things we're interested in is that there's this balance between these biological and chemical things that happen to control oxygen concentration, but there's also these physical things that matter. So like the strength of the wind and its ability to mix the water column and bringing in oxygen from the atmosphere into the water can be a major control on whether or not you have a hypoxic zone. And it turns out to measure all these different processes sort of over enough space and frequently enough to really understand what's driving what is really challenging. So one of the tools that I often use, and my colleagues do as well, are numerical models. And so we take kind of everything we know about the system, express it in mathematical formulations, and then link those mathematical formulations into a computer program which will then represent how the water moves in Chesapeake Bay, how the organisms move in Chesapeake Bay, and how the chemical reactions occur in response to various things like temperature and the availability of certain compounds. And so by using those models, we can run experiments, kind of what-ifs, to say if we reduce nutrients or cut off nutrients, what would the hypoxic conditions be? If something physically changed, or 1 year that had really high river flow from the Susquehanna River versus one that didn't, what would that have in terms of effect on low oxygen concentrations. So really that's sort of at the forefront of what we've been doing recently to try to understand this problem.